of factor D. Now the factor capital B, small b, as you can see here, this combines with the factor C3b. So these two components combine to form a new component that is called C3 convertase. You can see that the components of the C3 convertase are different here. In the alternative pathway, the component of C3 convertase are C3b and capital B, small b. But the components of C3 convertase in the classical pathway were C2b, C4b and C3b. Now this C3 convertase, it activates C3 itself. It causes more activation of C3. That is why this loop is also called as the amplification loop. The activation of this loop is promoted in the presence of bacterial and fungal cell wall. As we have told, the trigger for this pathway is endotoxins present in the bacterial or fungal cell wall. And this pathway is inhibited by molecules on the surface of normal mammalian cells. Now, as I told you, C3 convertase is formed and C3 convertase activates more C3. Also, after the formation of C3 convertase, the formation of C5 convertase takes place. Then, as we have seen in the classical pathway, C3 convertase activates C5 convertase, which causes cleavage of C5 into C5a and C5b, and C5b combines with other factors, the 6, 7, 8, 9 factors to form membrane attack complex. Okay. Now next, here in this diagram, there are two pictures here where you can see all the steps of alternative pathway. Next, the third pathway is Mannose binding lectin pathway. Specifically, this is sometimes called as only the lectin pathway. As it is suggested by the name here, mannose binds to lectin. Now, where is mannose present? Organisms like Neisseria or Candida, they have glucose and mannose residues in their cell wall. Here, a protein, mannose binding protein, capital MBL, mannose binding protein, which is synthesized in the liver, in our liver. It binds to the mannose residues present in the cell wall of pathogens. So, when bacteria enter our body, MBL recognizes them and it binds to them and it further activates MBL associated serine proteases called MASP1 and MASP2. What is the function of these serine proteases? They trigger the activation of C4 into C4a and C4b and also C2 into C2a and C2b. Of these components, the C4b component and the C2a component, they combine to form the C3 convertase. Now C3 convertase activates C5 convertase, C5 convertase forms membrane attack complex. So as you can see in all the three pathways, up to the formation of C3 convertase there are differences, and after that the pathways are similar. The end result is almost similar. But in the classical pathway, The end result may be either phagocytosis by opsonization. We have not talked about opsonization. What is opsonization? There are some proteins which are called as opsonins. The pathogens are covered by these opsonins. They mark them for destruction by our immune systems. So the components of our immune system recognize the opsonins present on pathogens and that marks them for destruction. This process is called as opsonization. Okay. So by the classical pathway, either phagocytosis through opsonization can take place, or lysis of the microbe can take place by MAC formation, or by mast cell degranulation and vascular destruction can take place. By the alternative pathway results in inflammation, which is leukocyte activation, adhesion, and chemotaxis. So the pathways differ in their trigger. The pathway differ up to formation of C3 convertase, and the pathways differ in their effector function. The final function may be a bit different, but overall they are bringing about innate immunity in the body. Now here is a chart which is showing functions of the components of alternative pathway. In this slide, you can see a summary of all these three pathways. Now, lytic pathway. Lytic pathway is the converging pathway. It is not a different pathway. It is just telling you that what happens after formation of C5 complex and C5 convertase and membrane attack complex. That is called as the lytic pathway. Now, these complements play an important role in our innate immunity. It plays a critical role in inflammation and defense against bacterial infection. Deficiencies of individual complement components or inhibitors of the system can lead to various diseases as shown in this table. The deficiency of few of the factors can result in certain bacterial infections or hereditary diseases. Here this is another comparative chart that is showing you the different steps and the converging steps. Okay, so here we have differentiated, in this chart we have differentiated between the three pathways, the different features of the pathways and what are the differences. Now the final thing here is functions of the complement system. What is the major functions of the complement system? The major function of complement system is lysis of infectious organism, activation of inflammation, opsonization, we have discussed opsonization, and immune clearance. Finally, immune clearance. In this slide again you can see the complement component deficiency, the deficiency of various complements. Okay. Here I have just noted down few of the questions that may come for your final exams. Okay, there may be a short note on MAC, short note on the diseases produced due to deficiency in various components, a note on the classical pathway or the lectin pathway or the alternative pathway. A 2-mark question can be on opsonization. What is opsonization? This is 2 marks. Another 2 marks can be what is a complement. Yeah, that's it. Here you can see few of the references from which I have studied and presented this topic to you. So, thank you very much.